science friends. I hope you all are doing well, that you've had a good week at home and have been able to rest a little bit and maybe enjoy some fun in the snow. Anyway, I wanted to take a minute to talk about some of the things that you all worked through yesterday. I'm not going to really give another assignment for today because what you need to do, you have to have some materials that I have at school. So we will work on that hopefully on Monday when I assume that we will return to class. So you've been working on lesson 2.2.5. You should have worked through these 2 pages yesterday. So yesterday you should have learned a little bit about meiosis and then some of the errors that can happen in meiosis, some of the mutations, and a little bit about non-disjunction. But I want to talk about those things, kind of some of the differences between mitosis and meiosis, and then also walk through non-disjunction a little bit also, because I know these things are hard to kind of figure out on your own. So we talked about mitosis when we discussed Kai. So remember, Kai had the tumors all over his body, and mitosis is how cells divide. It's how cells regenerate. So when you started as a tiny little baby or as a newly conceived baby, you were one cell. And so the way you got to as many cells as you are today is the process of mitosis. That one cell split into 2 cells, and then each of those 2 cells split into 2 more cells and more cells and more cells until you're this huge mass of cells now. Okay, now you're not the exact same cells that you started out with. Many of your cells have to be replaced over time. Maybe they get damaged. Some of them just have certain lifespans that they only last a certain length of time. And so mitosis is the process that not only helps you grow, but also helps you replace cells because they're damaged or just because they need to be replaced. So that is mitosis. In mitosis, you take one cell and you make 2 cells. And those 2 cells are not only genetically identical to one another, but they're identical to the parent cell that they split from. So the 2 new cells are called the daughter cells, and the one that they came from is called the parent cell. And those are all identical to one another. So if you remember mitosis, I'm going to write a little bit here, and you all can write some of this down as well. So mitosis, you've got I, PMAT, C. So you've got interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. And if you look at mitosis, that's meiosis. This picture is pretty similar to mitosis. In interphase, you're going to start to see the chromosomes form. In metaphase, you're going to see the chromosomes line up in the middle. Anaphase, you're going to see the chromosomes pull apart. And telophase and cytokinesis, you're going to see them separate. Now, what's slightly different between meiosis and mitosis is you're only going to have one of these sets of chromosomes. You're not going to have that second set here. These are homologous pairs or homologs, and you only see those homologs or homologous chromosomes. Those are in meiosis only. You don't see those in mitosis, okay? So let's talk just a little bit about these 2 processes. So let's talk about mitosis first. Now we're going to use human chromosome numbers. That's what we're talking about. So the parent cell in mitosis has 46 chromosomes, okay? In interphase, the DNA replicates, or the chromosomes replicate. And so then you have twice as many. So you have 92 chromosomes. 46 times 2 is going to give you 92 chromosomes. So that's after interphase. Okay, and then that cell in the process of mitosis is going to divide into 2 cells, and each of those would have 46 chromosomes. So they would be genetically identical to one another and also genetically identical to the parent that they came from. Okay, we've got I, PMAT, C. So I, the DNA replicates. P, you see the chromosomes become visible. M, they line up in the middle. Okay. A, anaphase, whoops, they pull apart. And then telophase, you start to see 2 nuclei. And then cytokinesis, the cytoplasm divides. So that's all of the steps of mitosis, okay? This happens in all of your body cells. Every cell but your sperm and eggs do mitosis. These cells are sometimes referred to as somatic cells. That's another fancy word for body cells, okay? Now, meiosis, oops, let me get my pen growing back up here again, okay? Meiosis, you're going to do I, PMAT C, which should look really similar to mitosis. And then you're going to kind of do it again, PMAT C. Okay? So this part is called meiosis 1. And this part is called meiosis 2. So in meiosis, there's 2 rounds of cell division. Whereas in mitosis, there's only 1 round. But there's only 1 interphase. So the DNA only gets copied once. Okay? So if we're drawing a cell for meiosis, you've got that parent cell. And it's got 46 chromosomes. Okay? It's going to get copied. So after interphase, it's going to have 92 chromosomes, okay? And then in meiosis 1, that's meiosis 1, it's going to split, so you'll have 46 and 46. And then in meiosis 2, they're going to split again. And so in meiosis, you get 4 cells, okay? And each of these are different from one another and different from the parent cell, okay? So they're genetically unique. They also have half of the number of chromosomes between the parent and the daughters. The daughter cells have half the number of chromosomes as the parent cells, okay? So meiosis forms reproductive cells, also known as your sperm and your eggs, okay? Now, think about this for a minute. Why is it important that sperm and eggs only have 23 chromosomes instead of 46? Well, the reason is you can take your sperm with 23 plus your egg with 23 and you get your zygote, which is your baby, that has 46. Had they have both started out with 46, the offspring that was made would have 92 chromosomes, which is way too many. You can't live with 92 chromosomes, okay? So meiosis produces cells with half the number of chromosomes that are genetically different from one another, and it makes 4 of them, okay? So that's very different from mitosis. As far as what happens in each stage, it looks the same. Metaphase, they're lined up in the middle. Anaphase, they're pulling apart. Telophase, you have 2 nuclei. Cytokinesis, the cytoplasm splits, okay? That's all in meiosis 1, okay? This is showing meiosis 1. If you get over here, then you have meiosis 2. And you go through the same things again. Prophase, the nucleus forms. Metaphase, they line up in the middle. Anaphase, the chromosomes pull apart. Telophase, you have 2 nuclei. In cytokinesis, you're going to end up with 1, 2, 3, 4 cells once those cytoplasms split. Each of these cells is going to have half of the number of chromosomes. See here, there's 3 little squiggly lines compared with over here, there were 6 little squiggly lines in the original cell. Okay, so it split the chromosome number in half. That's very important. Now, there's one special thing that happens in meiosis, and that's called crossing over. Crossing over happens during prophase 1. And crossing over is why the chromosomes, why every sperm and every egg is different from one another. Remember I told you that meiosis produces genetically different cells or genetically unique cells? One of the reasons why, the main reason why really, is because of crossing over. So here you have one of your these are homologous chromosomes, this set. Okay, so if we're looking at this is chromosome 1. You got one set from your dad, essentially, and one set from your mom. Okay, so this is chromosome 1. These are called homologs or homologous pairs. What happens is these homologs come together and they switch parts of themselves. And so as a result, you're going to have this blue has part of the red, and this red has part of the blue. You get what's called recombinant chromatids, okay? These are sister chromatids. Those two are sister chromatids. And these two are sister chromatids. These two, the blue and the red, are non-sister chromatids. So that's what crosses over, non-sister chromatids. The same colors are sisters. Different colors would be non-sisters. And so when these split apart and go to different cells, you're going to have one chromosome that's all red. One cell would have a chromosome that's all red. That would be like this guy here. You're going to have one chromosome that's mostly red plus a small amount of blue. And that would make up another cell. That's this one, here. You would have one cell that's going to get this one, mostly blue with a little bit of red. And then you have another cell that's going to get this chromosome, all blue. Okay? And so each one of these 4 are different. And you can see that in the picture here. These have crossed over. See how they have those little colored tips right there? And so if you look in the offspring, these are going to form 2 cells. And this only has 3 sets of chromosomes. So you have 26 pairs, or 46, chromosomes, 23 pairs of chromosomes. But see how this one is all red? This one's red with a little bit of blue. This one's all blue. But they're all 4 going to be completely different from one another. And that's what makes these cells genetically unique because of crossing over. Okay? So, differences between mitosis and meiosis. Meiosis is going to happen in reproductive cells. Produces what's called gametes. That's what we're talking about when we say sperm in your egg. Those are called gametes. Mitosis happens in body cells. We said those were called somatic cells. In mitosis, you have one division that gives you 2 cells. In meiosis, you have 2 divisions, meiosis 1 and meiosis 2, that gives you 4 cells. Okay? In mitosis, you have the same number of chromosomes between the parent and the daughter. So you start out with 46 chromosomes, and you end up with 46 chromosomes. Okay? This one, you start out with 46 chromosomes, and you end up with 23 chromosomes. If you have the full set, that's called the diploid. If you have only half, that's called the haploid number of that organism. So for humans, our diploid number is 46. That's how many is in all of our body cells. Our haploid number is 23. That's how many is in our sperm, our egg, our sex cells. Okay? Mitosis, the cells are going to be identical. Meiosis, the cells are going to be different. And then the events, you don't have homologous chromosomes in mitosis. You do have homologous chromosomes in meiosis. You have crossing over in meiosis. That is not a thing in mitosis. And I think that's really all. Okay? So about mitosis and meiosis, very, very important that you understand the difference between those two. Okay? You don't have to understand all the ins and outs and the processes and all the terms like sister chromatids and non-sister chromatids and homologous pairs, but you need to understand the main differences here, these kind of 5 main differences. You need to understand that mitosis makes genetically identical body cells and that meiosis makes genetically unique sex cells. Okay? If you got that, you're doing good. Okay? Sorry, I sound really hoarse and kind of gross. Now, the last thing that you read and talked about that you should have written just a little bit down in your binder is something called non-disjunction. So, non-disjunction can produce abnormal. It's a mutation. Okay. So what happens in non-disjunction is basically the chromosomes do not divide properly. And as a result, you will have cells like these that have too many chromosomes. And you will have cells like these that have too few chromosomes. Now, these are our gametes. These are our sperm and egg because this happens in meiosis, okay? Not in mitosis. So, non-disjunction is going to occur in anaphase. It can happen in anaphase 1 or anaphase 2, in meiosis 1 or meiosis 2. So basically, remember in metaphase, they're lined up in the middle. In anaphase, the chromosomes pull apart. But if they get stuck together, like they do here, that's non-disjunction. And as a result, normally each cell should inherit 2. This is what would normally happen. Those chromosomes pulled apart perfectly. And each cell should look like this. But look at these instead. This one has an extra set. Excuse me. This one has 1 too few sets. That's because of non-disjunction. That's because those chromosomes got stuck together in anaphase. Okay? So down here, these have too many. These have too few. That's it happening in meiosis 1. Over here, we see it happening in meiosis 2. So they pulled apart fine together in the first round, but in the second round, they got stuck together. So here, this would actually produce, this cell would go on to produce normal gametes because it was unaffected. This cell, however, would go on to produce gametes that have an abnormal number of chromosomes. And the term for this, when they have an abnormal number of chromosomes, is called aneuploidy. Okay? When you have an abnormal number of chromosomes, 1 extra or 1 too few. So Down syndrome is an example of this. Okay? Down syndrome is called trisomy. Whoops. I think we may have talked about this. Trisomy 21. That means they have an extra copy of their 21st chromosome. If you look at their karyotype, there will be 3 chromosomes there under the 21 number. Okay? And they got a cell, either a sperm or an egg, their mother or father, one of them, their reproductive cells did not divide properly, and their sperm or egg came out with too many chromosomes. And as a result, they had Down syndrome. Okay? So that's something that's a result of non-disjunction. So hopefully that clears that up for you all a little bit. You don't have any new pages to do in your binder today. Just make sure that you labeled these. This one should have been prophase, metaphase 1, anaphase 1, telophase or cytokinesis 1, and then prophase 2, metaphase 2, anaphase 2, telophase or cytokinesis 2. Okay, 3 differences in mitosis, meiosis. We kind of covered those here. I think those were also on PLTW. Non-disjunction, we talked about that. Now, these are different mutations that you can have, and those were walked through specifically in PLTW. So just look for this. There's some little arrows that you can slide through between these different pictures and write a description down of what that happens. So we talked about mutations on a gene, like when you get one extra letter, like maybe you're supposed to make CCG and you make CGCG, like you did an insertion there. That's a gene mutation, okay? These are chromosomal mutations, and they're going to have a lot greater effect because they potentially impact a lot more genes. Remember, chromosomes contain many genes. If you've got a chromosome, you know, it looks something like this, and there's genes here, genes, you know, there's genes all along that chromosome. And so if you delete a section of a chromosome, we're going to delete like 3 genes there. Versus if you make GUACGG, whatever, and you delete one of these, you're just going to affect 1 gene there. So this is a gene mutation. If you delete a section of a chromosome, that's a chromosomal mutation. This one would have greater effects because it potentially is going to affect more genes. Okay? I hope that makes sense. We can revisit this stuff in class when we're back together on Monday. Y'all have a great day and a great weekend. Bye.